Hello, everyone, and welcome to today's live broadcast, SRRF and TIRF, simultaneous spatiotemporal super resolution and multi-parametric fluorescence microscopy. My name is Christy Jewell. I'm from Labroots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by Labroots and sponsored by Andor. For more information on our sponsor, you can click on the sponsor logo on the left side of your screen or visit andor.com. Now let's get started. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many of your questions as we have time for at the end of the presentation. Also, please notice you can share today's webinar on your personal social media. Just click on the social sharing tab to let your friends and colleagues know about today's live event. Now, if you have any trouble seeing or hearing today's presentation, just click on that support tab found at the top right of your presentation window, or you can use that Ask a Question box to let us know you're experiencing a problem. I would now like to introduce today's presenters. Thorsten Wohland, Professor of Biophysics, Department of Biological Sciences and Chemistry at the National University of Singapore, and Dr. Alan Mullan, Product Specialist for Microscopy, Cameras at Andor Technology. For complete biographies on our speakers, please visit that biography tab at the top of your screen. Gentlemen, welcome. You may now begin your presentations.
Hello and welcome to this webinar on SRRF and TIRF, simultaneous spatiotemporal super resolution and multi-parametric fluorescence microscopy. My name is Thorsten Wohland and I'm from the National University of Singapore. Experiments in this work have been performed and evaluated by Hari Krishnan Balasubramanian and Jagadish Sankaran in my group. Jagadish and Waiho Tang from Adrian Röhlin's group have established the GPU algorithms, and Sonia Monti is a scientific illustrator who interned in my lab and helped with some of the graphics. Well, the good ones anyway. Typically, fluorescent measurements are performed and optimized with a single evaluation method in mind. However, in principle, the data contains much more information if it can be mined with multiple different evaluation approaches. Here I'd like to show this on the example of spatial and temporal resolution and how we can maximize the extraction of information from an experiment by optimizing acquisition parameters. Let me motivate this and explain why we even need both spatial and temporal information. Take a look at the picture of Newton sitting under the apple tree. Which of the apples was the most important for his discovery of the principles of gravity? The apple on the ground, the apple hanging on the tree, or the apple falling? Of course it was a falling apple, which just shows how important dynamics are. Take a look at the next picture. This shows fluorescently labeled peptides binding to a bilayer. But only if I switch on the movie and you see the peptides moving do you clearly see the bilayer and its holes. It's the dynamics of the peptides that show you the structure of the bilayer. We need to record the structure of as well as the dynamics in a sample to obtain maximum information about the system. But this is not everything, and we could be also interested in the aggregation state of the peptides, or whether the peptide is just bound to or inserted into the bilayer. Therefore, we should aim to apply multiple spectroscopic techniques to the same data set to optimize information extraction and answer all these questions simultaneously. Let's first talk about some limitations of fluorescence imaging that make this task difficult. First, we need light sources, nowadays typically lasers, for the excitation of the sample. Concerns here are stability, the power we have at our disposal at a particular wavelength range, and whether the source can be pulsed or modulated. With modern lasers, this is not much of a concern anymore. The second factor are the optical systems for the collection of the fluorescent signal. The main problem here is detection efficiency. Large fractions of photons are often not collected in current setups, even with the most modern of instruments. The next factor are the fluorophores that provide our signal. We possess a wide-ranging set of fluorophores, from organic dyes to fluorescent proteins to semiconductor nanocrystals. Here the concerns are fluorophore brightness and photostability, but also the strategy to deliver and label proteins of interest. As labeling is of paramount importance, genetically encoded fluorophores are often still the fluorophore of choice despite their non-optimal fluorescent properties. Therefore, we have to often adapt our techniques to best exploit the information provided by fluorescent proteins. Finally, we come to detectors. Here we have point detectors, for example avalanche photodiodes, APDs, or photomultiplier tubes, PMTs, or array detectors that can consist of APDs and PMTs, or of course scientific CMOS and EMCCD cameras. Important issues here are the quantum efficiency, that is the amount of photons detected, the time resolution and readout speed, and the overall signal-to-noise ratio. So overall, especially the probes and detectors determine our signal-to-noise ratio, as we have only limited numbers of photons per molecule and limited detection of these photons. In addition, of course, the number of photons detected depends on the time delta t and the area of A squared over which we record the fluorescent signal. And this brings us to the following problem. A particle creates an image in our detector of the size of the point spread function of the optical system. To capture dynamics, we need to acquire at least one photon in 2 consecutive frames. Therefore, our acquisition has to be fast enough to capture the particle at least twice. This requires on one side sufficiently large pixels and on the other side sufficiently fast acquisition. To capture the position of a particle, we need small pixels to resolve the PSF and be able to localize it. We need to capture the signal of the particle on at least 2 but ideally more pixels for any localization. The uncertainty in our localization is described by the following equations, where r is the radius of the point spread function. If the particle moves, that will actually increase. A is a pixel size, n is the number of detected photons, and b is a background signal. So for good localization, we need small pixels and slow acquisition to maximize the number of photons n. So at first glance, spatial and temporal resolution seem to be mutually exclusive. As the number of photons depends on both acquisition time and pixel size, it seems that we can't optimize both. To record molecular dynamics, we use large pixels that allow us to acquire the signal fast and still obtain sufficient signal levels. This results in low spatial but high temporal resolution. To reach super resolution, we need small pixels resulting in much lower acquisition to reach the same signal level. This results in high spatial but low temporal resolution. So these two approaches are not compatible, and approaching the problem this way, we cannot reach simultaneously high spatial and temporal resolution. Note that in addition, the problem is confounded by the fact that the number of pixels recorded determines the time one needs to read out a frame, and thus this also limits time resolution. The solution is simple. We just record small pixels at high frame rates. Afterwards, we bin either in space or in time to optimize the data for super resolution microscopy or the different spectroscopy techniques we would like to apply. However, this mode of measurement became only possible with advances in camera technology and data evaluation strategies. Modern cameras can now read out with high detection efficiency and signal-to-noise ratio, and they can read out even small pixels at high frame rates. These are now sufficient to observe the movement of single molecules. And the advent of GPUs allows us to evaluate the data of thousands of frames collected per second in real time. So let me introduce very quickly the optical setup that we used in this particular study. It consists of a commercial total internal reflection fluorescence microscope, a wavelength splitter that allows to record 2 different wavelength images on a single camera, and a fast and sensitive EMCCD or scientific CMOS camera. In our particular case, the TIRF setup is advantageous as it illuminates only a very thin slice of the sample above the cover slip. It thus avoids background from the bulk of the sample and has high signal to background and high signal to noise ratio. In the last years, we have combined different spectroscopic techniques measuring dynamics. This includes a combination of imaging fluorescence correlation spectroscopy, spatial and dual color fluorescence cross-correlation spectroscopy, single particle tracking, and fluorescence recovery after photobleaching. Here we will combine imaging fluorescence correlation spectroscopy with number and brightness analysis and super-resolution radial fluctuation microscopy. Typically, these measurements are performed in separate experiments or even use different instrumentation. Here I'll show that we actually can obtain the same information from a single dataset. In the following, I'll first explain the three techniques and provide some ideas how to perform the data evaluation. In imaging fluorescence correlation spectroscopy, or imaging FCS, we image the movement of fluorescent particles on a camera. The crossing of particle through a pixel will lead to fluorescence intensity fluctuations in that pixel. The fluctuations contain information of how the particles move and how many particles there are, so about the particle's diffusion coefficient and concentration. Evaluation of the autocorrelation function of every single pixel provides this information via the width and the amplitude of the correlation function. Longer fluctuations mean slower particles and produce wider correlation functions. But as fluctuations are more difficult to see at higher concentrations, the amplitude of the autocorrelation function is inversely proportional to the concentration, meaning that higher concentrations result in lower amplitudes and lower concentrations in higher amplitudes. We can depict the diffusion coefficient and the average number of particles seen by a pixel in maps. As an example, I show here the fluorescently labeled lipid rhodamine PE in a phase-separated bilayer. The diffusion coefficient map shows areas of fast and slow diffusion, while the concentration map shows areas of high and low concentration. The concentration map coincides with the TIRF intensity image, but the diffusion coefficient map is just the inverse. This is the case as rhodamine PE will be trapped in areas where it moves slowly and has a higher concentration in these regions. This analysis can be performed on an image stack by a plugin for Fiji or ImageJ called Imaging FCS, and which is freely available on our website. In the next slide, I'll show a short video how to install the Imaging FCS plugin and how to use it. To install Imaging FCS on your computer, please start Fiji, go to Help, and Update. After a short time, the ImageJ updater window will appear. Then please go to Manage Update Sites and look for Image Science. Click Image Science and Imaging FCS. Close and apply changes. Downloading of the files will take about a minute or so. Click OK. Quit ImageJ and start again. Now let's take a look how to operate the plugin. You'll find the Imaging FCS program under the plugin menu. Imaging FCS 1.52 being the current version. When you start the program, it will automatically detect whether there is a compatible GPU on your computer. If not, it will run in CPU mode. In my case, on my laptop, I don't have a GPU and run on CPU mode. Click on the Load button and look for files you want to analyze. I've prepared here already a file. It's a phase-separated bilayer. Open, and the program will automatically tell you how many images you have in your stack. In this case, we have exactly 20,000 frames. Now proceed to put in your experimental parameters into the panel. This consists mainly of the frame time, which is in our case 1 millisecond, the pixel size of your camera, which is 24 micron in our case, magnification and NA of your objective, and the emission wavelengths of your sample. The phase-separated bilayer has been labeled with rhodamine PE, and thus our emission wavelengths is around 580 nanometers. To calculate autocorrelation functions, you can click now anywhere within the image. In the dark part, you get very good autocorrelation functions as you see. This is a liquid part of the bilayer. The bright part is a gel phase, and here the correlation functions look very different. If you want to calculate multiple autocorrelation functions at the same time, choose a region of interest and click ROI. You'll get now all correlation functions at the same time. To fit the data, click on the Fit button to obtain the Fit window. The Fit window allows you to use different models by selecting parameter values and selecting whether they are free or are to be fixed during the fit. In this case, we'll fit only the number of particles, that is the amplitude of the correlation function, and the diffusion coefficient of one component. Again, click anywhere in the window. You see now the correlation function in blue and in red the fit. We have now 3 new windows. We get the residuals of your fit. We get a window which will contain the map of the parameters in your image and a histogram window with a distribution of parameters. If you want to obtain the correlation functions and their fits for the whole image, please click All. Now the program will calculate all correlation functions and fits. It will open as well a log window where it will tell you if there were any problems in fitting any of the functions. Here you see now all correlation functions at the same time. You see that we have two different kind of correlation functions. Good correlation functions from the fluid phase and the less well-defined correlation functions from the gel phase. You have here now a parameter window, and you see here now as well the distribution. The first map is the number of particles. You see that wherever we have a large number of particles, we have bright regions. Where we have a low number of particles, we have dim regions in the TIRF image, as expected. If we switch over to the map for the diffusion coefficient, we see exactly the opposite. Wherever we have a large number of particles, we have low diffusion coefficient, that's a gel phase, and wherever we had low number of particles, we have very fast diffusion. The correlation between the number of particles and the diffusion coefficient can be explained by the fact that in the regions where the molecules move slowly, they will spend more time. Thus, you accumulate a large number of particles in the gel phase, while in the fluid phase, the rhodamine PE moves very fast and does not accumulate. If you want to review particular autocorrelation functions, you can just select a region in the map, and then you get only the correlation functions with their fits corresponding to that particular region. If you go to the dark area, you get the correlation functions for the gel phase. This is now only a very basic overview of the imaging FCS program. If you want to know more about all the parameters in the panel as well for the fits, please go to the documentation you can find on our website. Thank you very much. In number and brightness, or NNB analysis, we again look at the fluorescence signal in a single pixel. If we define the number of particles in the pixel as n and the brightness as epsilon, then the average fluorescence signal is given by the brightness times the average number of particles. The variance of the signal will be given by the average of the brightness times the fluctuation in the number of particles squared. Since epsilon is a constant, this is equal to epsilon squared times the average of the squared fluctuation signal. The number of particles seen by a pixel follows a Poisson distribution. Therefore, the variance of n is equal to the average number of particles n. With this, we can define now the average fluorescence signal as epsilon times n, and the variance of the fluorescence signal as epsilon squared times n. So we arrive at definitions for the average number of particles and brightness for our number and brightness analysis. Since cameras are not photon counters, these values have to be corrected for camera offset and noise. And finally, one has to take care to eliminate shot noise contributions to the variance, which can be done by calculating the covariance of neighboring intensity values. As has been shown by Unruh and Grattan, this provides good estimates for NNB analysis as long as the time between measurement points is much smaller than the time a particle remains in the pixel. The problem, however, is that we need a good calibration sample to determine the brightness of monomers and dimers. For this purpose, we have created monomer and dimer constructs of fluorescent proteins that are bound to the plasma membrane over a so-called plasma membrane targeting sequence as shown here. This task is complicated by the fact that not all fluorescent proteins are fluorescent. Therefore, even the dimer constructs comprise molecules with none, single or both fluorescent proteins being fluorescent. It can be shown, however, that the ratio of the brightness of the dimer construct compared to the monomer construct -1 just represents the probability of a fluorescent protein to be fluorescent. This assumes that the fluorescence of the two proteins is independent. We have performed these measurements for the fluorescent protein EMBL. we used various exposure and measurement times to determine what optical acquisition parameters provide stable NB values. On the left and middle graphs, you see the monomer and dimer brightness for PMT and MAPL, for measurement times between 20 to 100 seconds, and for 3 different exposure times per frame of 2, 10, and 20 milliseconds. On the right side, we depict the dimer-monomer brightness ratio. It is evident that although the values do not vary strongly, they stabilize at a value of about 1.5 for measurements longer than 60 seconds and exposure times longer than 10 milliseconds. According to these measurements, the exact probability of a MAPL to be fluorescent is 53%, which is quite good for a red fluorescent protein. Measurements in the rest of the webinar were all evaluated with 20 millisecond exposure and 100 second measurement time. In the next slide, I show a very short video of how to perform NMB analysis in the Imaging FCS plugin. With the Imaging FCS plugin, you can also perform number and brightness analysis. This we typically do at longer frame times, 20 milliseconds in this example. For the number and brightness analysis, we first need a background file that has the same dimensions and the same number of frames as our actual file. Press on the background button and select the background file. Now press on load to select the file you want to analyze. We have here a CHO cell expressing PMT-MAPL, which is a membrane-bound fluorescent protein. Let's take a look at the image stack. There are 2 things to realize. First, intensities outside the cell are typically below 2000 counts. Intensities on the cell are way above 3000 counts. So to analyze only pixels on the cell, we can set an intensity threshold of 2700 counts. Secondly, you realize that if you look at the stack, that the sample is bleaching. Therefore, we will use a bleach correction based on polynomials of 4th degree. Now you can go to number and brightness analysis. We will do a G1 analysis as explained in the webinar and press NB. The program will warn you that you have less than 100 data points for the last correlation channel, which is fine. After about a minute or so, you'll see 2 new windows appearing. The blue one will be the information about the number of particles. That's very similar to the autocorrelation analysis we've done before. The yellow one will show you the brightness values. Repeating the analysis for monomeric and dimeric fluorescent protein constructs allows you to calibrate the system and determine how many of the fluorescent proteins are actually fluorescent. Then you can continue and analyze your sample of interest. Let me make a small but important remark. As indicated earlier, the collection of thousands of frames per second results in large amounts of data that have to be evaluated, ideally in real time. CPUs are not capable of performing these tasks, but with the advent of GPUs, we can parallelize and accelerate calculations for number and brightness analysis as well as imaging FCS. The 2 plots here show the time taken to evaluate an image stack as a function of the number of pixels per frame. The plot on the left is for imaging FCS autocorrelation function calculations and data fitting, the right plot for NB analysis. In both cases, GPU analysis is faster, starting with about 100 pixels per frame, and from about 1000 pixels per frame, the GPU is about 20 to 40 times faster than the CPU. For example, the full imaging FCS evaluation of a 128 x 128 image stack, including data fitting, takes around 36 seconds on a Titan XP GPU, but it takes 20 minutes on a state-of-the-art i7 CPU. Let's look now at the biological question we'd like to address. The epidermal growth factor receptor, or EGFR, is a prototypical receptor tyrosine kinase that has been studied over many years. Nevertheless, there are many unanswered questions about its activation mechanism. It is, for example, still debated whether EGFR exists in dimeric form before ligand binding or only once ligand is bound. Second, Although there is evidence that EGFR interacts with the cytoskeleton, it is not known whether these interactions significantly influence receptor mobility and oligomerization. Here we aim at answering these questions. Does EGFR exist as dimers before ligand binding? And does it interact with the cytoskeleton? To answer these questions, we need to have accurate knowledge where the cytoskeleton lies under the plasma membrane and whether at these positions the receptor mobility is modulated or whether it oligomerizes. In other words, we need high spatial and temporal resolution. Let's start with NMB and its calibration. On the left you see TIRF images of PMT and EMBL monomers and dimers. In the middle you see maps of the NMB analysis of the constructs in CHO cells. As can be seen from the brightness values, the dimer has a much higher value, although it is not double of the monomer brightness. If you plot the histogram of the brightness values, it also becomes apparent that the PMT and EMBL dimer is brighter than the monomer, although the distributions strongly overlap. If you perform the same measurements on EGFR and EMBL in the absence of ligand, we get a brightness that is slightly lower than that of the dimer. We see the same if we plot the EGFR brightness histogram, in black on the right. The histogram lies somewhere between the red monomer and the blue dimer curve. Further analysis shows that this can be explained if at least 63% of EGFR is in a dimeric state, in line with earlier findings by us and other groups. Actually, even higher oligomers cannot be strictly ruled out. After demonstrating imaging FCS and optimizing NMB analysis, we now come to the optimization of SRRF, the computational super-resolution method used here. The advantage of SRRF is twofold. First, it is readily available via an ImageJ plugin that runs on GPUs. Second, as it is computational, we can optimize our spatial and temporal binning to optimize the data for SRRF. That said, note that any other computational super-resolution or deconvolution algorithm could be integrated as well. SRRF has been explained in another webinar in detail, so let me just recapitulate the basic concept. First, SRRF divides existing pixels into subpixels with equal value. Then it determines in radial direction whether intensities increase or decrease toward that pixel. Therefore, it determines whether the source of the intensity is likely to be located at that pixel. Second, it makes a correlation analysis to determine whether the intensity is related to noise. Noise would show no temporal correlations, while a signal will have a correlation fingerprint. This way, we end up with structures whose location is now better defined than in the original image, and that can serve as a basis for the comparison of dynamics and structural data. Let's take a look at the cytoskeleton in CHO cells. For this purpose, the acting structures were labeled with LiveAct EGFP, an acting binding protein. All images were taken with TIRF at 2 millisecond time resolution. The pixel size in object space was 120 nanometers. On the left you see the raw TIRF image and a selection of intensity cross-sections across actin fibers. In the middle we have SRRF applied to these images with SRRF pixel size set to 1/5 of the actual pixel size, that is 24 nanometers. At the bottom you see the cross-section across actin fibers at the same position as in the first case. In the third graph, on the right, we repeated the SRRF analysis, but now after binning the frames in time to 20 milliseconds. One can see that the fiber diameter as measured by the intensity cross-section decreases, allowing better localization of the fiber. Extending the SRRF analysis to 200 millisecond time binned frames brings down the feature size to about 60 nanometers. Just as a control, we have performed the same analysis with 2 times lower magnification. As expected, in this case, the feature size determined from SRRF is of course larger, but also potentially creates artifacts which we will talk about later. For the rest of the webinar, we use 200 millisecond time binning and 120 nanometer pixel size in object space for SRRF analysis. Now let's talk about the dynamics measurements by imaging FCS on the same dataset. First, we have to make a difference on how and where the dynamics are measured. In the schematic here, you see an acting fiber as it is seen by TIRF in green and as it is seen by SRRF in cyan. We can measure the dynamics at different binning either on the fiber, at places between fibers, or just at the border between the regions. If we use 2x2 binning, the signal-to-noise ratio might be lower, but it also samples a smaller area and will provide more detailed diffusion coefficient data. 4x4 binning has a higher signal-to-noise ratio, but it will measure not only the fiber but also off-fiber regions at the same time. Thus, it will be harder to differentiate between fiber and off-fiber regions. To illustrate this, I will show in the next figures only two cases, namely on and off fiber measurements and at 4x4 binning. Correlation functions of 4x4 binned pixels that are located directly above visible actin fibers provide a diffusion coefficient of about 1.4 micrometers squared per second. Regions between fibers, which we label here as off-fiber, show a more irregular shape consisting of two parts. First, a fast decaying correlation function at short times is marked by the red square indicative of diffusion. Then they show slowly varying artifacts at long times, which could be related to aggregates in solution or low amplitude sample movement that is only seen at small correlation amplitudes and low signal-to-noise ratio. If we concentrate on the fast decaying portion of the correlation function, which we show here normalized to emphasize the similarity of the decay, we obtain a diffusion coefficient of about 2.9 micrometers squared per second. This indicates that diffusion close to the fiber is slower than diffusion away from the fiber. A reason for this could be the binding and unbinding of live actin to the actin fibers. Can we improve on that? First, let's decrease binning and refit our data. With 2x2 binning, each observation area becomes smaller. The areas between fibers have now so low signal-to-noise ratio that they cannot be fit anymore. These pixels are indicated in white in the diffusion coefficient map. Pixels on the fiber are now better defined and show a much smaller diffusion coefficient of 0.77 plus minus 0.53 micrometers squared per second. The lower diffusion coefficient results from the fact that the pixel is smaller and now sees mainly the fiber and little area around. To improve on that even more, we can use the SRRF map and use it as a mask for our diffusion coefficient map. So we will now take only those pixels in the diffusion coefficient map into account that have a SRRF identified fiber below. This changes the diffusion coefficient only little, but reduces the standard deviation, providing overall better precision of our parameter recovery. But this is not the end of the story. Let's select on the unfiltered diffusion coefficient map a region around a fiber as shown in the green box. Now we can select autocorrelation functions for pixels directly on top of the fiber according to SRRF and pixels right next to the fiber. We can see in this plot that the autocorrelation function on top of the fiber has a higher amplitude and thus higher signal-to-noise ratio. If we plot the number of particles, that is the inverse of the amplitude, we find in all locations that the highest amplitude, that is the lowest n, is on the fiber. Therefore, the FCS data corroborates the SRRF data, and the SRRF data can be used to refine and select the FCS data. We can go one step further and use FCS data now to remove artifacts from the SRRF data. On the left you see the TIRF image. If you carefully look inside the yellow box, you see some very weak fluorescence intensity in there. On the right side you see the SRRF corrected DMAP. SRRF clearly identifies the points 1 to 5 in the yellow box as valid structures. Let's take a look at the correlation functions at these points and compare them to correlation functions found on fibers in other areas of the image stack. This slide is a bit busy, but if you compare the correlation functions on the top row that are from the 5 points identified earlier with the autocorrelation functions in the bottom row that were taken from clear fiber areas, you can see that with the possible exception of point 1, all other autocorrelation functions on the top row have a lower amplitude and signal-to-noise ratio than the correlation function on the bottom row. Therefore, we can identify at least 4, if not all 5 points as artifactual and not representing real fibers. With this, we can now mutually filter the SRRF image and the DMAP to refine our dynamics measurements and remove SRRF artifacts. Note, however, that this of course does not bring FCS beyond the diffraction limit, but it improves FCS data. This is a demonstration of the validity of the approach and how dynamics and structural data complement each other. But the real strength of the approach lies elsewhere. Namely, in multicolor recordings. Here we show image stacks in which the cytoskeleton was again imaged by live ACT in the green channel. We applied SRRF to the green cytoskeleton channel as before. In the red channel, we record the epidermal growth factor receptor labeled with MAPL. The red channel was analyzed by imaging FCS as seen in the purple graph and with NNB analysis as seen in the yellow graph. As we have now collected all these parameters from the exact same data set, we can now compare the parameters at each pixel. When plotting a scatter plot of the brightness versus the diffusion coefficient, we saw to our surprise a negative correlation. This means that brighter and thus larger EGFR oligomers diffuse slower in the membrane. As membrane proteins do not follow the Stokes-Einstein relation and should have a very weak dependence on size, we hypothesize that the larger particles are pre-internalization aggregates and are in a different environment. It should be noted though that if we plot brightness against the SRRF map, there is no correlation, meaning bright particles are not in general found close to the cytoskeleton. Finally, if we plot the diffusion coefficient against the SRRF map, we also do not see any correlations. This indicates that EGFR diffusion is not directly hindered by the cytoskeleton. As there is evidence that the cytoskeleton influences EGFR diffusion, we assume that this action is indirect and it is the influence of the cytoskeleton on the lipid membrane organization that then changes EGFR diffusion. I hope that these two studies convince you that there is more to be had from your image stacks. Dynamics and structure are two sides of the same coin. They are complementary and can inform each other, improving structure determination and dynamics parameter estimation. When performed in multicolor experiments, parameter correlations can be established that are not available from sequential measurements. This combination of microscopy with spectroscopy measurements can truly provide new biological insights, as has been shown before for confocal setups. But here, now one does not need any specialized instrumentation, but just needs a fast camera and any existing TIRF microscope. With this, let me summarize the webinar. We acquire data at high speed with small pixels. We bin in time or space to optimize the data for various techniques, that can be FCS, NNB, SRRF, etc. The structural and dynamics information can be used to error-proof each other and obtain better super resolution and dynamics data. GPU implementation brings data treatment even for large errors into the minute range. And we have shown structure-dependent live-ACT diffusion. Finally, for EGFR, we have shown that diffusion is related to oligomerization, but not directly to the cytoskeleton. In the future, we are working on a direct camera readout with simultaneous data evaluation. If you want to have more information on the ImageJ plugin for imaging FCS, please go to our website. In the end, let me thank again all the group members who were directly involved with this work. Jagadish and Hari for all the measurements and evaluations, Jagadish and Waiho for the implementation of the GPU algorithm, Daniel for work on direct camera readout, Sonia for the help with the graphics. And thank you very much for listening.
Thank you, Thorsten. Hello, I'm Dr. Al Mullan, a product specialist for microscopy cameras at Andor Technology. And before we move on to the questions for the main part of the webinar, I'm just going to look at some of the detector solutions that were mentioned that would be available for super resolution and FCS studies. So as we've seen, detectors play a role as being one of a number of key enabling technologies that we need to help study the inner workings of the cell, so that's the cell structure and dynamics as well. And in terms of imaging cameras, we can group these together into EMCCD and SCMOS technologies. EMCCD cameras have long been used for super resolution from D-Storm, PAM, STED and so on, as well as being heavily used for FCS experiments. But in more recent times, there have been more sensitive SCMOS detectors, and we have seen these appearing and being widely used in other super resolution techniques such as SIM as well as DNA paint, just for a couple of examples. And these detector technologies and the super resolution and FCS techniques are explained in more detail in the Andor Learning Centre. So I'm just going to cover just very briefly some of the main imaging detectors available and how they may or may not suit the different super resolution and FCS experiments. EMCCD detectors have been the detectors of choice for a long time for whenever the utmost sensitivity and detection of the weakest signals are required, and this is due to this unique on-chip amplification that amplifies the signal many-fold. So this does not eliminate read noise, however it allows the signal to be boosted many times before it is finally read out. And unfortunately you don't get anything for free, and the downside of this is this can introduce additional noise whenever we're dealing with higher signal levels. And the serial nature of this readout process in CCD cameras ultimately limits the speed that they can run at. In terms of current EMCC detectors, we have two examples. One is a 1/4 of a megapixel with a large 16 micron pixel format. An example we just mentioned for this is the Andor Exxon Ultra 897, and the largest available, which is a 1 megapixel 13 micron pixel format, and the example of that is the Andor Exxon Ultra 888. And EMCCD has been around for quite some time, but over the years it has been refined, and we would see the ultimate versions of these in the Ultra and the slightly lower cost Life series. And also just to mention about SurfStream. SurfStream is a live cell real-time implementation of SURF, which Thorsten mentioned earlier. SCMOS detectors can be referred to as active pixel sensors because the charge to voltage conversion is performed on the pixel itself, and then it is read out on a line-by-line basis rather than a pixel-by-pixel way that it is done in CCD. So this allows for a much faster readout process. And in terms of sensor developments, most of the recent sensor developments have been with SCMOS, although like with CCD cameras, CMOS has been around for a very long time indeed. So with the SCMOS cameras, models now use the back-illuminated technology that had previously only been in the CCD scientific cameras, and this allows the quantum efficiency to be boosted from originally 60 through to 72 to 80, and now through to above 90, reaching up to 95%. But with SCMOS, because we have a lot of pixel-to-pixel amplifiers and differences potentially between them, we do have to consider how any very slight pixel variations may need to be considered depending on the super resolution or the correlative fluorescence measurements that we may be making would have. And for SCMOS cameras, we can look at two examples, both from G-Pixel, and these are both 4.2 megapixel sensors, and one of them has a larger 11 micron pixel, and the example for that is the Sona 4.2B-11, and the one with the 6.5 micron pixel, which is the Sona 4.2B-6. And the Andor Sona series of cameras, this is really something that was developed to try and extract the very best potential from these new sensors that are now available. The first thing we need to consider with detectors is their sensitivity. They need to be sensitive enough to work with the signal level that we have, and often we can have a weak signal from whatever number of fluorophores we're working with, and in real life having to capture this against a background. We can also have short exposures. We can be looking at blinking fluorophores, trying to capture dynamic events within the cell, or measure very small fluctuations in signal intensities. And for live cell super resolution, we need to try and keep illumination intensities as low as we can in order to achieve as accurate cell biology as we can. And if we're thinking about trying to make use of the data, we would be thinking of getting better data in captured by the camera to allow us to get better data out to allow us for any downstream analysis that we need to do. And as Thorsten mentioned, there's some other things we need to consider that relate to this, such as pixel size, with smaller pixels providing better sampling but less photon collection efficiency. But then we can also use pixel binning to try and improve signal to noise, but the downside of that is we will lose spatial resolution. If we look at the theoretical signal to noise ratio plots, we can see that with the latest generation of back-illuminated SCMOS cameras, they're providing a better signal to noise ratio than EMCCD, right down to somewhere below the 10 photons per pixels mark. And of course, this is going to vary depending on your microscope configuration, because you may use additional magnification, for example, which might impact the effective pixel sizes, just for one example. So actually, in reality, we're still finding that many experiments still require the sensitivity of EMCCD. So this is not just limited to single molecule studies. We are still finding quite an interest in EMCCD cameras, and ultimately for the really sensitive work they're still required. But whenever we don't need this level of sensitivity, then we can start to consider the back-illuminated cameras and look at the advantages that we know that we can get from SCMOS technology. So now then that we're considering the back-illuminated CMOS cameras, we can look at both the speed and field of view of the options we have available. So in these two options, we can see that they're both 4.2 megapixel cameras, but of course we're going to get a much larger field of view with the one with the 11 micron pixel size. So this will also potentially allow us to have a better signal to noise ratio because we have a much larger photon collection area available to us. However, the other model with the smaller pixel should be able to allow us to have better spatial resolution. And in terms of speed, we can see in these examples here that the Sona 4.2 B-6 is a faster camera, although the other Sona model is still sufficiently fast for many applications. And whenever we crop the sensor, we can boost speeds much higher. We can also see here with the EMCCD cameras, they are still capable of quite high speeds, up to 100 hertz or frames per second at smaller regions of interest. So for these cameras then we can see that the Sona with the GS400 BSI sensor is providing the largest field of view and is still fast enough for many applications. And this is the largest area SCMOS with a large pixel, and that means that it will suit high magnification such as 100x, and when sensitivity is more important than resolution or having the highest speed. With the other Sona model with the GS2020 BSI, it is a faster sensor to begin with and with a smaller pixel size. It also has a useful correlated multi-sampling mode that allows it to have a lower read noise, and that drops the noise floor even lower, which is very important for whenever we're dealing with very low level signals, as ultimately they are going to be limited by the read noise. So this particular camera, the Sona 4.2 B-6, will provide a balanced performance of sensitivity, high speed, resolution and field of view, and it will also have the better sampling, which is going to be useful for different things, whether that's deconvolution or localization based super resolution, because it's always useful to have improved sampling of the image. And this also makes it suited to the more common 40 and 60x objectives according to meeting Nyquist criteria. And in terms of the Sona model itself, or the Sona camera platform series, this has been developed from experience of both the Exxon EMCCD cameras and the previous generations of SCMOS cameras in order to extract the best performance from demanding applications such as single molecule, super resolution, and fluorescence correlation spectroscopy. So some of these include being able to access the full sensor. The Sona 4.2B-11 has a 62% higher field of view than other cameras using that same sensor, and uses an exclusive anti-glow technology to avoid sensor glow and access up to 32 millimeters field of view. These cameras also use the permanent vacuum technology, which is normally only seen in the Exxon EMCCD cameras, and that allows us to have superior reliability and cooling performance, and allows us to reach the lowest noise floor. And because these sensors actually have quite a high dark current, this is quite important to apply moderate levels of cooling. And this also helps then where we need to have slightly extended exposures. It allows us to have a wider window of operation with these sensors. Another effect of the cooling the sensor that we can do with this camera is that we also make benefits in terms of sensor uniformity and requiring less pixel corrections, and this can be quite important for making localization or correlative measurements where we are relying on a uniformity from one pixel to another. Higher quantitative accuracy is another thing we took great care on, and this the SCMOS cameras, such as the Sona and also the Xyla models, have a very high linearity of over 99.7%, and this allows for more accurate and precise measurements of fluctuations in fluorescence, and this is going to be important whenever we're relying on these to make quantitative measurements. And another thing that we looked at as well was to do with minimizing vibration within the camera, and that is done in a number of ways, and we also have water cooling as an option. So that again is another way to eliminate issues with the vibration impacting sensitive measurements and having any impact on trying to localize a signal. So sometimes only an EMCCD camera will do. It's just not possible for even the latest SCMOS cameras to operate down to signal photon per pixel levels, and the Exxon Ultra 888 will provide the largest possible area and speeds from EMCCD. So whenever we don't need that ultimate sensitivity, the back-illuminated SCMOS cameras like the Sona series are going to prove to be a much better solution for super resolution or FCS. So we can make use of the smaller pixels and take benefit of the improved sampling, improved spatial information, and potentially get some better localization accuracy as a result of that. We can also get higher frame rates, higher speeds, and better dynamic range as well. And the Sona models have been optimized really to deliver and extract the best from those latest sensors and get the best accuracy and precision possible from quantitative measurements such as used within super resolution and FCS experiments. So there's a wide range of super resolution techniques out there, so experimental requirements as well are also going to vary quite a lot. So it's always recommended to contact one of your local and/or representatives to discuss whatever may be your specific requirements. Okay, thank you. So now we can move back to questions for the main part of this webinar.
Thank you, gentlemen, for your informative presentations. We will now start the live Q&A portion of our webinar. Now to our audience, if you have a question you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, let's get started. Our first question is, let's start with this one. What is the best pixel size for FCS and SuperRes?
Yeah, that's an interesting question. So when we did FCS alone, we typically worked with about 240 to 400 nanometers in sample space. But that, of course, doesn't allow you to do super resolution. For super resolution, you'd like to be on the 100 nanometers or below. So for SURF and FCS together, we compromised. We stayed at 100 nanometers, and then we can actually bin 2x2 or 4x4 pixels to get good FCS data.
Thank you, Thorsten. Now, my next question is two parts. What are the advantages of brightness analysis by NNB than FCS on the imaging FCS instrument? FCS can also determine the brightness using average fluorescent intensity and number particle. And how do you correct differences of sensors in the camera? Probably differences of sensor sensitivity in the camera are directly affected to the resulted brightness.
Yeah, that's a very good question. So in principle, FCS can determine brightness as well, because ultimately it's just the amplitude of the FCS curve and the intensity you use. In confocal, that works very well. On cameras, however, the amplitude is not a good measure for the number of particles. So if you want to have a good idea about the number of particles, you have to calibrate your camera with a standard solution of known concentration. For us, it's easier to calibrate the brightness of a fluorophore than to try to calibrate the number of particles in a standard solution, especially if we work on membranes, because we don't have very good membrane samples where we know the concentration very well. What one could do compare confocal microscopy with imaging FCS, but then this makes it more complicated. The calibration of the fluorophores is much easier with NNB. That's one of the reasons. How do you correct differences of sensors in the camera? Now, we calibrate, because we have the constructs already, we calibrate actually every sensor with these constructs. So, and you have to use a background file for each camera, because each camera is going to have a slightly different noise pattern. So you need that to correct your data. But overall, what you saw on the number and brightness analysis, the values are actually very constant once you calibrate it.
Thank you, Thorsten. Now, we have so many questions that we're not going to be able to answer. I'm going to squeeze a few more in, but those questions we can't get to today during our live Q&A, they will be answered via the email address you provided at your time of registration. So let's try to squeeze a few more in. Does this work with light sheet microscopy?
Yes, actually it does. So we have done this in the past as well. We have published on that. The difference between TIRF and light sheet microscopy is the light sheet is, of course, much thicker than the TIRF field. TIRF is only about 100 nanometers thick. So that's actually ideal for membrane proteins or for membrane measurements. Since we do a lot of membrane measurements, we work a lot with TIRF. But we use light sheets, for example, in zebrafish. We have measured morphogens, morphogen dynamics in interstitial spaces or in the membrane in zebrafish with light sheet FCS.
And do we need anything special for our experiments?
Well, you need a TIRF microscope or light sheet microscope. Otherwise, you need the camera. And you need a software that allows you to do the correlations or the number and brightness analysis, depending on what you want. Otherwise, the technique works with organic fluorophores. It works very well with fluorescent proteins, as you've seen here. We almost use exclusively fluorescent proteins in cells because it's much easier to label for us again. So that's all you need.
And speaking of labels, do we need special labels?
Well, yeah, so as I just tried to indicate, of course, organic dyes give you better signal-to-noise ratio. So we have beautiful, nice curves on artificial bilayers. But the moment you go into a living system, a cell or a small organism,
it is much easier to use fluorescent proteins as labels. And we have used a wide range of different fluorescent proteins from mCherry to EGFP. So they seem to work all quite well. And they give the same results as we typically get with FCS, confocal FCS.
Thank you. And we'll wrap with this final question, and we'll go with this one because it's come in a few times. What about dual color FCCS?
Yes, that can be done as well. We have at the moment shown it only with TIRF. So we have measured, for example, the EGFR dimerization, which we have shown here with NMB. We have shown that before as well with imaging FCCS. We are working at the moment to do this as well with a light sheet microscopy.
Thank you, Thorsten. Now, before we go, I do want to thank our audience for joining us today and for their interesting questions. Again, there are quite a few questions we are unable to answer. They will be answered via the email address that you provided. I would like to once again thank Thorsten Wohland and Dr. Alan Mullan for their time today. We'd also like to thank Labroots and our sponsor Andor for underwriting today's educational webcast. This webcast can be viewed on demand. Labroots will alert you via email when it is available for replay. In that email, there will also be a downloadable link, a link where you can download your certificate of participation. We encourage you also to share that email with your colleagues who may have missed today's live event. We thank you for joining us. Stay safe, and we hope to see you again soon. Have a great day.
Thank you very much.
Thanks, everyone.